Have you ever had a piece of chocolate that just stops you in your tracks? One that's so good you remember it for weeks? Well, it turns out the secret behind that unforgettable experience isn't just about the cocoa beans or the recipe. It's a microscopic drama, a wild party of microbes that makes all the difference. So let's get into the incredible science of what makes great chocolate great. Right? We've all been there. You have one piece of chocolate and it's a flavor explosion. It's complex, it's floral, it's got a story to tell. And then you have another piece and it's just brown, it's sweet, it's fine. So what's the secret ingredient? What's the real difference between amazing chocolate and just chocolate? Well, in the chocolate world, experts basically split it into two big categories. On one hand, you have bulk chocolate. This is your standard, everyday cocoa flavor. It gets the job done. But then you have fine flavor chocolate. This is the good stuff, the prized stuff. We're talking complex notes like flowers or caramel, way less bitterness, and an aroma that's just incredible. The big mystery has always been, how do you get that fine flavor result every single time? It all comes down to a critical step that happens right after the cocoa beans are harvested. It's called fermentation. Basically, farmers pile the fresh, gooey beans into boxes and just let them sit for about a week. Now, if you skip this step, you don't get anything that tastes like chocolate. You just get something intensely bitter and, well, gross. Fermentation is where the magic starts. It's where all the potential for that amazing fine flavor is born. But here's the catch. On most farms, this whole process is completely spontaneous. Microbes from literally everywhere, the air, the pods, the workers' hands, the wooden boxes, they all just jump in. It's a completely wild, unpredictable microbial party. And since you never know who's going to show up, the results are all over the place. Sometimes you get a fantastic batch, and sometimes not so much. So to figure this all out, a team of scientists decided they needed to become detectives. They went straight to the source, cocoa farms in Colombia, to conduct a full-blown forensic investigation into this microbial mystery. Their mission? To finally crack the fermentation code. And they really did treat it like a crime scene. They were on site at these farms, meticulously collecting samples from the fermentation boxes, taking all sorts of measurements and gathering every single clue they could find to understand exactly what was going on inside that microbial party. A typical fermentation goes for about 7 days, so 168 hours. At key moments, like at the 48 and 96-hour marks, the whole pile of beans gets turned to mix in some air. Our science detectives were there the whole time, taking samples at each critical stage to build a complete timeline of the entire process, from start to finish. They were hunting for two main types of evidence. First, the physical clues, the abiotic factors, things you can measure, like temperature and pH. And second, the biological clues, the biotic factors. This was the who of the investigation, the actual microbes throwing the party. Okay, their first physical clue was a big one. It was the temperature. They saw that inside the box, the temperature didn't just stay the same. It rose in a very distinct S-shaped curve. And that was a massive hint, because that curve perfectly matches the classic growth curve of microbes. It meant the microbes themselves were working so hard that they were literally generating their own heat. The second clue was pH, and this was interesting. It was like a tale of two different worlds. The sugary pulp on the outside of the beans became less acidic over time. But at the same time, the inside of the bean, the part that actually becomes chocolate, called the cotyledon, did the complete opposite. It became more acidic. This was proof that there were really complex, separate chemical reactions happening all at once. So, with the physical scene set, it was time to look at the biological fingerprints. And what they found wasn't just some random mashup of microbes. Oh no, it was a highly coordinated, dramatic succession, a full-on turf war where different families of microbes took over in very predictable waves. In the first 24 hours, the bacterial scene was completely dominated by one major family, Erwinaceae. These guys were the first ones to arrive at the party. They were feasting on all the easy-to-get sugars in the pulp and just absolutely running the show. But fast forward to 48 hours, and the whole scene had changed. Those early partiers, the Erwinaceae, were on their way out, and a new boss was in town, the Acidobacteriaceae family. They loved the new environment that the first wave had created, and they took over for the rest of the fermentation. It was a clear 1-2 punch. And get this, the same kind of drama was happening with the fungi. Early on, it was a big mix of different types. But as things progressed, yeasts from one super dominant family, Saccharomycetiaceae, which includes the famous yeast we use for baking and brewing, rose up and totally commanded the later stages of the party. Okay, so the detectives had all their clues lined up. They had these predictable patterns in temperature and pH, and this coordinated microbial takeover. But the million-dollar question was still hanging in the air. Does any of this actually connect to how the final chocolate tastes? I mean, that's what it all comes down to, right? They've got all these microbial fingerprints and all these physical clues. But does any of it really matter for the final flavor? Could all this data actually predict a delicious, complex chocolate bar? Or was it all just, scientific noise? The answer was a huge, resounding yes. When they crunched the numbers, the links were incredibly strong. For example, the presence of a specific fungus called Turlispora was directly linked to those beautiful floral notes. And our hero yeast, Saccharomyces, was tied to those delicious caramel and panela, that's a kind of unrefined sugar, flavors. They had found them. They'd found the actual flavor makers. So finding the flavor makers was one thing, but could the scientists actually take control? Could they assemble their own microbial dream team to recreate that fine flavor magic on command every single time? This was it. This was the ultimate test of their whole theory. And they believed they could do it because of a key scientific idea called functional redundancy. That sounds complicated, but it's pretty simple. It just means that in that huge, wild microbial party, lots of different microbes can do the same essential jobs. You don't need the whole crowd, you just need to make sure all the critical tasks are covered by at least one member of your team. So they came up with a 3-step plan. First, they mapped out the genes of all the microbes they found to see what chemical tools each one had. Second, they identified every single essential chemical job that had to get done to create those flavor precursors. And third, they handpicked the smallest possible team of microbes that together could do every single one of those jobs. So how big was this microbial dream team? After sifting through that entire world of cocoa fermentation, they narrowed it down to just 9. An elite squad of 9 strains, 5 bacteria, and 4 fungi that they believed could do the complex work of hundreds. They called it their synthetic community, or SynCom for short. The team was assembled. The theory was solid. Now it was time to run the experiment and see if this hand-picked microbial crew could actually deliver the fine flavor goods. So, what was the verdict? And what could this mean for the future of chocolate? The results were, frankly, stunning. They had a panel of trained professional tasters try chocolate made with the SynCom. And the scores, well, they speak for themselves. The chocolate made in the lab with the Dream Team scored right up there with a world-class fine flavor chocolate from Madagascar. And it completely blew the standard bulk chocolate out of the water. They did it. They had actually reproduced fine flavor in a controlled lab setting. This discovery just opens up such a fascinating future. Think about it. If we can control the microbial guest list, does that mean we can guarantee exceptional flavor in every single batch? Could we finally move past the random, unpredictable nature of fermentation and into a new era of designer chocolate, where every single bar has the potential to be truly unforgettable? The science seems to be telling us that that future might be closer than we think.